What I love about our field is that it bridges stem cell biology and neuroscience. And that's been fascinating to understand more how the brain works and how it can change.
Originally from Canada, Fiona Doetsch has been based in Switzerland since 2014. She works at the Biozentrum in Basel, a centre entirely dedicated to molecular life sciences. In her laboratory, on the 12th floor of this building, inaugurated in 2021, she studies stem cells in the adult mammalian brain. Most of these stem cells are quiescent, meaning they remain in a dormant state.
In the brain, there are stem cells which are dormant, so they're not actively dividing, they're metabolically active, but they're constantly sensing signals in the environment. And they either stay quiescent or they sense a signal that activates them to begin to divide and then give rise to progeny.
While the existence of these neural stem cells is now recognized by the scientific community, it was long overlooked. For more than a century, it was widely accepted that mammals stopped producing neurons shortly after birth.
At the time, the idea that new neurons could be added seemed completely counterintuitive, because how can we maintain memories throughout life if our neurons are constantly changing? So with the discovery of new technologies that allowed us to better follow these cells and really ask the questions, it was possible to show that these were happening. But then finding the stem cells was also an exciting journey because nobody expected glial cells to be stem cells.
Fiona Doetsch has indeed demonstrated that physiological stimuli can regulate different stem cell pools to generate specific types of neurons, as well as glial cells, which form the neuronal environment. These findings were made possible by studies on the mouse brain. The work was carried out in 3 dimensions, using a technique developed by Fiona Doetsch's team.
This is a model of a mouse brain, and I'm going to open it up. And here in yellow, you can see the stem cell niche. And in green are the new neurons. They migrate as these long chains all the way forward to the bulb. Many of the projects that we have in the lab now, we actually use these whole mount preparations to see cells that are sitting on the surface of the ventricles and to get a global perspective at once. You can see everything that's happening through the entire wall in one snapshot, instead of having to reconstruct serially through millimeters in the brain.
Through precise and rigorous research, Fiona Doetsch and her team have shown that certain neural stem cells in pregnant mice are activated to generate new neurons. Neurons entirely dedicated to the sense of smell, enabling mice to recognize their offspring.
The amazing thing is that the cell type that is newly generated doesn't stay there, but has to migrate in a completely different location. And this is even baffling for people in the neuroscience field, trying to understand that these cells actually physically move and migrate to the olfactory bulb, which is the center for the olfaction and the smell. This was the first time that we see these neurons actually being involved in a very important behavior for the mouse.
There's a lot of course, brain activity and hormones and other things that regulate behavior. But what she really showed is that the development of new cells in the brain also influences this behavior. So this is conceptually very, very interesting and important.
In the past, people used to think that brain plasticity was largely due to remodeling of the connections in the brain or the synapses, which is very important. But here, we were showing that new neurons were being added and functionally important in a short manner. And this is because certain sets of stem cells were turned on. So stem cells really then are an essential part of brain plasticity.
Many of us who study the brain are sort of stuck into thinking only about one type of cell, which is the neuron. And Fiona, with this sort of attention, with this focus on the other half of the brain, really pushes us to sort of have this holistic view of brain function and not get stuck only in one specific niche, if you want, which is the neuron type.
She's an excellent scientist, an excellent teacher. She can really catch people and fascinate them for biological topics. We need role models like Fiona.
Today, Fiona Doetsch and her team continue their work to better understand this neurogenesis in the adult mammalian brain. How and why these neural stem cells become activated and what signals they respond to are among the many questions explored daily in her laboratory.
We are interested in the heterogeneity of the stem cells in the brain. What makes them different?
If you look the cell from the cover, they're all the same. However, actually they're all different. And our idea is to understand, how they respond in different physiological states.
When a cell is in the tissue, it receives a lot of signals. It's not only one signal that will make everything happen. So there is a balance between signals that they receive, these neural stem cells receive from their environment, microenvironment, but also intrinsic signals.
So kind of a dream question, it would be, to kind of isolate sets of signals coming from different elements of the niche and how they together recruit a set of stem cells. Not all, just a set. But this is like a long-term question.
While there is still a long way to go to fully decipher these complex mechanisms, Fiona Doetsch's discoveries open up new perspectives and could have major implications for both fundamental biology and medicine.
It is not too far-fetched to think that understanding the basic mechanisms of how the brain is able to create new neurons and integrate into existing circuits will allow us to engineer, in a certain sense, this type of process to maybe even stimulate endogenous production of neurons or supply from the outside world.
I think in the long term, it's very interesting to see whether the brain might have some capacity for self-repair. And then also, because of our findings with different physiological states, perhaps just by lifestyle changes, for example, exercise or other sort of natural interventions, it may be feasible to eventually stimulate these different stem cells for brain repair.